So, so we left off our topic on this exact space, right? We wanted to talk about, we were talking about the Golgi apparatus, right? And I want to make sure that you guys understand the reticulums and the Golgi apparatus, and it's very easy to go back and read about them and be able to absorb the information really well, right? So now we need to talk about a very important topic which comes up again and again, right? And I want to make sure this lecture is holistic. That means if you're reading a topic, you should also be able to understand it well enough, what you not miss out on it, right? That's how a lecture is supposed to be different from a book, because in a book, if you don't go to chapter 12, for instance, where they're teaching about protein, you're missing out on it. But right now, I want to make sure you're not missing out on it. And this one slide will sum up the entire process for you. So it's self-explanatory. There is a nucleus. Again, appreciate the double membrane and the nuclear pore, from which there is a obviously endoplasmic reticulum. Guess which one is this? This has dots on it. It's obviously rough. Those dots are obviously protein ribosomes. You can see that, for example, let me take this one ribosome over here. It's getting some amino acids which are being converted into this long protein. That protein makes its way through the cisternal space towards the end of the endoplasmic reticulum, and through this endoplasmic reticulum, it must have been modified into some structured chain structure. And even if it is not modified into a changed structure, let's say three of these were packed together, the rough endoplasmic reticulum, using its brain, obviously, pun intended. The nucleus is the brain of the cell, and the RER uses its mechanism to put three of these together. It senses that these three are, for example, their hemoglobin. Now you remember hemoglobin needed 4 chains, right? And so it puts together those 4 chains. Or for example, if this one is, let's say, collagen, needs those 3 chains. It puts them together in one vesicle that buds off, enters the cis phase of the Golgi complex somewhere far off. That cis phase is going to put all of these collagen together, cut their corners to get the perfect piece. Those perfect pieces at the Golgi will be modified slightly. For example, calcium will be added to them. Certain enzymes will work on them to make them hard if it's collagen. And eventually the final products will be packed together in secretory vesicles that will then leave the Golgi apparatus, bud off from here, and go combine with the cell membrane to release those proteins on the outside. The outside can just be the tissue. The outside can be the bloodstream, right? Hormones can be released right into the bloodstream. Similarly, hormones have a lipid part. So guess where it's being made? In the smooth endoplasmic reticulum. So that's essentially it. And this slide is absolutely wonderful in explaining the relations, right? Now we need to move on to the next organelle, which is the mitochondria. Now, again, we're going to always, whenever we're starting, we're going to start with talking about the integrity of the cell membranes, right? So this mitochondria has, again, a double membrane. There is a single membrane on the inside, which is this folded membrane, and then a membrane on the outside, which is the smooth membrane. Mitochondria is special in the sense that the space between these, because the folded membrane is obviously more inwards, right? The space between the inner and the outer membrane is the intermembrane space, which is very, very important. You'll see the importance of this in the Krebs cycle and you making ATP, but essentially it's a double membrane organelle. It has a matrix inside, which is this water-like fluid. It's different. Why is it being called the matrix? It's different from the cytosol. Remember that first cell with the 3D cell with the water in it? That was cytosol. This is the matrix. The matrix has its own proteins. It has its own DNA. It has its own ribosomes. Now, how is that possible? You're having one DNA in the nucleus and another DNA or multiple DNA in those many worm-like mitochondria inside, right? Inside one cell. That is so strange, right? And that is a symbiotic relationship, in fact. It's a theory that mitochondria were essentially something like a bacteria that ended up in an animal cell, It helped the animal cell survive by giving it ATP, which is what led to us being this large in size, right? Otherwise, we would have been one of those, according to the theories, which we're going to also talk about in the ecological part of the topics. We would have been creeping prototists or something, but that symbiotic relationship really helped us become these huge, smart, beautiful human beings, right? So always remember, mitochondria is having its own DNA, and it has its own ribosomes. Its ribosomes are different from the ribosomes of an animal cell. An animal cell has, let's say, 80S ribosomes, right? Mitochondria is closer in structure to a bacteria than it is to us. So it has smaller ribosomes. Remember, smaller mitochondria, smaller ribosomes, 70S ribosomes, right? And then its own DNA with no nucleus. So few things that need to be put straight out here. Now, if it has its own DNA, then it's making its own proteins. It has a double membrane, so that ensures no protein goes out into the cytosol. It functions as a completely different thing within the cell. How is it symbiotic? It receives all the nutrients of the cell to make food for itself. That food it makes into ATPs, right? Those ATPs it makes in so much excess amount that it barely ends up using them and makes them in so much amount that they're released into the cell, and that cell then stays alive by those ATPs, right? So that's how the mitochondria essentially functions. So we need to understand it in a more in a more 2D view, because it's important to see what it's essentially like. So let's start from this diagram where we see an outer membrane and then a highly folded inner membrane, right? These inner membrane are highly folded. And remember that that worm, it had inner zigzag structures. Those zigzag structures were the foldings of the inner membrane, right? And it has its own DNA that is not packed in a nucleus, right? It has its own ribosomes that are 70S, its own proteins that are different. So the next thing you need to understand when you come to a mitochondria is that the outer membrane is a normal membrane regulating the inflow and outflow. The inner membrane is special. It has something called an F1 particle or an ATPase generating particle. It has, so the inner membrane is folded because remember, whenever you're studying the villi or the stomach or the hair cells, you always study, okay, there's higher foldings because you want to increase the surface area as compared to the volume ratio. You want to, in a small area, if you make a lot of folds or add a lot of smaller particles instead of one large block, it increases the surface area. And that's exactly what happens. It's highly folded inwards to increase surface area where more and more F1 particles can be attached, right? These F1 particles are protein, obviously protein particles that are only made by the DNA of this mitochondria, right? And they're attached to the membrane. They help in generating ATP, right? This is an electron microscope. You can see how the mitochondria looks like. Look at the outer membrane and then the inner membrane. Look at how deeply folded it is. And then there's there's no nucleus, there's DNA molecules lying around. So this is an electron microscope because the details of this membrane are better viewed under the electron. And you look at these are ribosomes floating all around because this is a cell that's highly functional, making lots of proteins. So what is ATP? It's essentially energy currency of the cell, right? ATP is like a battery. It's a charged battery. It releases energy to form ADP and an inorganic phosphate. And then this ADP and an inorganic phosphate comes inside the mitochondria to this F1 particle. This F1 particle is like the charger. It gets charged again to give you more ATP, right? So this charged battery comes from that F1 particle. The next structure, like the mitochondria, is the chloroplast or something called the plastids. The chloroplast, so let's understand what the chloroplast is. The chloroplast, again, double membranous. It has intermembrane space, just like the mitochondria. The only difference is it's only present in animal cells, not in plant cells. It has coin-like stackings. Imagine these as coins being stacked on top of each other. These, these stacked coins occur in another fluid called the stroma, just like the matrix. This here is called the stroma. A stacked pile is called a granum, and inside these cells, these coin-like structures, this yellow fluid is our chlorophyll or any other pigment it might be. You know, it can be red, chlorophyll is green, so it can be red pigment, pink pigment, whatever pigment possible, right? And these cells, each of them is covered by a membrane known as the thylakoid membrane. So that's essentially it. And you need to understand now what the plastids are. So plastids are essentially double membrane organelles, right? They're present in plants and algae. Their purpose is to manufacture food. How do they do that? So you know, chloroplasts absorb sunlight, use it. There's chlorophyll or pigments in here. Just like that, in plastids that have maybe, let's say, are of red color, it's going to be a similar structure with red pigment here instead of the green chlorophyll, which is going to trap sunlight. And that sunlight is then going to be used to make food through, let's say, photosynthesis. All of those processes that we did as children, right? So that's that for us. Then we have vacuoles or other vacuole-like structures such as peroxisomes or glyoxysomes. So you need to understand that vacuoles are single membrane lined. Now these structures are not double lined. There's one single layer. Imagine a circle. There's a single layer around it. Let's see if I can find an example here. So look at this. It's a vesicle, or a vacuole is similar. There isn't two membranes. This is the first and the second membrane of the Golgi, right? Vesicle only has one circular membrane, so single membrane. And that single membrane contains proteins. These proteins can be stored molecules, for example, or can be glycogen or anything to become storage vesicles. It can be enzymes to become vacuoles that are digestive, and so. Certain vacuoles can be given names such as peroxisomes and glyoxysomes, and there's a couple of others, but these are the important ones that, let's say, come up the most, right? Peroxisomes are an important integral part of cells. Why? Because they have oxidases and catalases. These peroxisomes essentially break down a molecule that is produced in a cell as a byproduct called H2O2. And remember, H2O2 is made as a byproduct, which is highly toxic. It's essentially bleach. You cannot have bleach in your cell. So what does a peroxisome do? It senses, okay, there's H2O2 being produced here. It's a reaction that produces H2O2. It will go and attach to that reaction, release glycolytic enzymes such as oxidases and catalases to break down H2O2 into H2O plus O2, which is water and oxygen, which is useful for us, right? That's what a peroxisome does. And then we have glyoxysomes. Now, glyoxysomes again are specialized vacuoles that have, or vesicles that have that are present in the plant cells. So they have their own special enzymes. Similar. Now, those enzymes are again catalases and oxidases. So whenever the word xisome or xisome comes in, it's a word that has catalases and oxidases. In glyoxysomes, these catalases and oxidases essentially occur in seedlings that are rich in lipids. They break down lipids or fatty acids, just like peroxisome was breaking down H2O2. And this is specific to seedlings, especially seedlings with a rich supply of lipid, to break that lipid down and release energy. Very simple. That energy is in a specific, so essentially this last line here, now don't get confused. I want to make sure my slides are easy. So I'm explaining every single word, right? Just what does succinyl mean? Succinyl is just a carbon, it's just like, you know, it's let's say if three fatty acids combine together, they form something called a succinyl. So it's like a very simple fatty acid. A normal fatty acid will have 2 million carbon atoms combined together. Succinyl will only have 2, right? So that's just the name for it. When there's 2 million, it's so difficult to use energy from them. When it's only 2 molecules, it's so easy. So that's essentially what a succinyl is. It's just a basic structure of a fatty acid. Many succinyls join together to form a long chain of fatty acids. That's essentially our topic on biological molecules, which we're going to also do. But yeah, you need to remember glyoxysomes are rich in lipid-rich seeds, okay? So lysosomes can be of those types, but then what if they're intracellular lysosomes, lysosomes that are supposed to work within the cell, right? Not secretory vesicles. They can have enzymes that have a digestive role as well. For example, your lysosome was released from your, look at this, it looks like a Golgi apparatus, right? Released from your Golgi apparatus, the preformed enzyme, it's so fresh, it's coming right from the source. It combines with, for example, your cell ate a food particle, a glucose molecule. That primary lysosome, when it combines with this glucose molecule, it forms a digestive vacuole. It's now going to digest the food particle. After digesting it, it's going to become a secondary vacuole. It's a secondary lysosome, right? Make sure you understand a primary lysosome is when it's fresh, a secondary when it's reacted with the molecules it's supposed to react, and then eventually so the enzymes are, so why are these enzymes trapped in lysosomes? Because enzymes are also self-destructive. What do enzymes do? Enzymes break down, for example, proteins, and enzymes themselves are made out of proteins, right? So after they finish eating up this material, they're going to eat themselves up, right? And that eating up of this molecule of these materials or everything like this is eventually going to lead to residual substances being left behind. These are useless. They're essentially, let's say, if the water and oxygen, if H2O2 is produced in this case, then a peroxisome will come running, attach here, and digest that H2O2 into oxygen and water, which is then exocytosed or released from the cell towards the outside, right? So endocytosed comes in, exocytosed goes out. And essentially lysosomes can, become digestive, or they can break down, they have enzymes, right? Enzymes can do anything, they can break down anything. They can, for example, if a virus particle comes in, they can attack that virus particle and break that down. Or your mitochondria or your other lysosome or anything else in your cell is becoming old, then what will happen? They will then attack that old cell and form another vacuole, right? Or another lysosome, just like a digestive vacuole or a digestive. So essentially, lysosome is that essential molecule. When a vacuole forms, when you have anything other than the enzymes also added to the cell, single surrounding cell membrane, which forms the lysosome, right? So if it has molecules from your own cells, it's called autophagic, which means auto, which means myself. So autophagic is called, and phagy means eating, right? So phagy means eating and auto means myself. So it's autophagic. Or if it's, for example, eating a bacteria, then it's phagocytic. Cytic means breakdown. So phagocytic, eating and breaking down something else, is a phagocytic vacuole. And again, the residual, the waste products will be exocytosed, right? So it's very easy. Primary lysosomes are fresh made, they're small, they have those inactive enzymes that are ready to get activated when they combine together with another vacuole or another similar endosome. Endosome is essentially anything that comes in, a vesicle that comes in after endocytosis, right? So this is undergoing digestion, and the residual products can be eliminated. So again, the table is very self-explanatory. The structure, the functions are very easy. We've already done most of them. So there's autophagy, there's phagocytosis. Again, autophagy is eating up your own organs. Phagocytosis is eating up anything from the outside. Degeneration, if your organelles have gone bad, new organelles need to be made. So the cell is continuously making the new organelles, right? But who's going to dispose of the older ones? Or can you just release them outside? No, they can be poisonous. They can be toxic to the outside world. So they're broken down by these autophagic lysosomes, right? And then these lysosomes can release enzymes for digestion of molecules inside, for example, food particles to form digestive vacuoles, right? Just like on the previous slide. Now, you need to understand a pathological point of view that is written in your book. So let's just make it very simple. Lysosomal storage diseases are the pathology. Pathology means disease. So in a lysosomal storage disease, what will essentially occur? The lysosome enzyme will not be working, right? So, like I said, the cell is continuously taking in food, making new organelles when old ones are going bad. And the lysosome is responsible for breakdown. But if the lysosome is not functioning, if the enzymes are not functioning, what will happen? All of that bad stuff will be stored in you. So it's called a storage disease because all of the bad organelles, everything that has gone bad is now not being broken down, right? So large molecules cannot be broken into smaller molecules. They cannot be removed. So they're being deposited inside the cells. And once they are being deposited, they're not only, so they're also taking up space, but they're also using your ATP. Now, if there's a mitochondria that cannot make ATP, but is only using ATP, now it's useless for us. The lysosome should attack and kill it. If the lysosome is not attacking and degrading it, all of those extra mitochondria will add up. And if not in, let's say, in the first year, if it does not add up, within after 1 year, there's going to be a lot of mitochondria. Then you will have signs and symptoms. And essentially, that's what ends up happening. In a lysosomal disease, it's basically a storage disease. All of those things end up getting stored in your body, and then it's essentially because it's always congenital. What does congenital means? It means when you are in your mummy's womb, that's when it starts. Your enzymes are malfunctioned right there, which is why your enzymes no longer function. The lysosome does not have active enzymes. Lysosomes are present, but the enzyme is not active. Then you can have buildup of those substances. Now, will a child being born have that disease? No. Initially, you'll think the child is normal. But then let's say 6 months later, after you started feeding him glucose, all that glucose could not be broken down. It was stored. It's just an example, does not necessarily have to be realistic. So all of that glucose or all of that star energy is not being broken down. It's just being deposited in the cells. Now, after 6 months, that person is going to have problems. He's probably not going to be healthy because he can't break that glucose down. He's probably not going to have good immunity and other things. For example, there's a Tay-Sachs disease in which lipid is not broken down, fats are not broken down. Now, what will happen? They will accumulate. So that person can be obese, but internally problems can occur. For example, cells may burst, cells may not be able to tolerate the problem, right? Similarly, breakdown of glycogen can be interrupted in something called the Pompe disease. So there's multiple diseases, and I don't know why I've made so many mistakes on this particular slide, but yeah, you have to go with the flow now. Okay, so that was our topic till the organelles, right? Now we have another organelle, which is the cytoskeleton. Cytoskeleton is a really interesting organelle. To understand the cytoskeleton, you need to understand that there's three kinds of cytoskeletons. And before going on, let's use this diagram. So all of these wiggly parts inside their cytoskeleton, right? Like I said, cytoskeleton helps attach one cell to another cell. Look at these. It's extending beyond the cell to help it anchor. It attaches the cell to the basement membrane or the ground as well, so that all cells are not floating around. Your cells are supposed to sit down. They're supposed to perform their functions stationary, not disrupt other cells, right? They're supposed to the organelles within the cells are transported by the cytoskeleton. So it's very easy stuff, book stuff, but you need to be able to imagine the cell. Imagine that I've only stained the cytoskeleton of the cell, right? And then there's one important structure. You can see that the cytoskeleton, the red is holding the nucleus in shape. The blue is probably carrying stuff, which is why it's so widespread and going around. This one is anchoring. What is this? Why has this been highlighted? It is called the MTOC. Let's try to understand what the MTOC is. Let me tell you, the MTOC is the control center. So the MT, I'll tell you what the MT stands for, but the OC stands for the operating center, let's say, right? This MTOC is responsible to control how this cytoskeleton behaves, but a specific cytoskeleton. So there's three kinds of cytoskeletons. There's a very small one called an actin filament. There's a very large one called the microtubule, and that MTOC comes from here. MTOC stands for microtubule organizing center. So this is like the father, the bab of the microtubules, right? So actin, very small, microtubule, very large, intermediate, like the word says, in the middle of both of them, right? So let's try to understand these filaments. Again, essentially screenshot this. It's an important diagram, very interesting diagram. So you need to understand that this diagram is explaining multiple things. For example, the structure. Actin filaments are made up of actin subunits, like the word says. Microtubules are made up of alpha and beta tubulins, and they occur in dimers. One alpha and one beta is one subunit of a microtubule. Intermediate are made up of keratin, vimentin, or lamin, right? Anyone, or probably other proteins as well. They're rope-like, and they're in the middle of both of these. So the measurements are 7, 10, and 25. You can see 7 is so small and 25 is so large, right? Actin are just two strands of this actin protein running side by side. Microtubules occur in a similar structure where these dimers line up, and 9 of these make one hollow tube, right? And then everything else, the functions are so they're very interesting and very important to explain at the same time, right? So actin, for example, maintain the cell's shape. So this is the important part. The actin is responsible to maintain the shape of the cell, right? So over here, look at these red ones. They're the actin. Look at these attachments between each other. They're the actin molecules. So these are maintaining shape of the cell. Then we have intermediate. These give the cell a solid shape. Now the actin are just maintaining the shape, but intermediate are rope-like. They give the cell a solid shape. They anchor the nucleus in place, anchor the organelles in place. Then we have the microtubules, the most important ones, the ones that you always study in details. So the microtubules are the ones that form the flagella or the cilia. Remember that tail-like thing on the bacteria? That was a microtubule, right? Remember the hair cells on the epitheliums? They're the cilia, right? So don't forget, microtubule form, they're the larger ones, and they form the larger tail-like things or the hair-like things. Flagella or cilia, always microtubules, right? They obviously help in shape, but they're like tunnels. These tunnels are responsible for moving organelles, right? So this is another important thing. Moving organelles or intracellular transport. Why? By forming tracks or tunnels, right? And just like their function of moving stuff, they have an important function during cell division. That is moving chromosomes. And how do they move those chromosomes? So remember that MTOC, the microtubule organizing center, these are giving out microtubules radiating outwards that will control how the chromosomes move. They move to opposite ends, and it's a very beautiful diagram showing you how this is a hollow tube with the dimers, right, forming the centriole. The centriole is the MTOC. Yes, the centriole is the microtubule organizing center. It forms something called spindle. That spindle is essentially what moves the chromosomes apart, right? Remember, there were chromosomes that aligned in mitosis and then they're pulled apart to different cells, right? So the thing that's pulling them apart is called spindle. So spindle is essentially just a microtubule. So it helps divide the cell into two. And the specific thing that occurs is that this MTOC or centriole moves to opposite ends of a cell from where it extends its microtubules that are going to pull the chromosomes apart and help in nuclear division or cell division or any other word that you'd like to use. So that's essentially what these cells are about. And finally, we have cell wall. You all know that cell wall is an integral structure, not in animal, but in plant and other kinds of cells. It's made up of cross-linking. Its cell wall is essentially dead. So you need to understand this. Cell wall is not a live organ. It's which is why I never wrote it organelles. I've written it. It's a dead organ, right? It's a dead part of the cell. It's deposited on top of the cell membrane. So if this was the cell membrane, these deposition of strands is forming the cell wall, which is why, because it's just a deposition, it can be thick in some places and thin in other places. It can be irregular. And because it's a dead part, it's just deposition, it does not have a regulatory mechanism of what enters or leaves the cell, right? So that's important to understand. So for example, and then each cell controls what its cell wall is made up of. For example, plant cells will deposit only cellulose, never anything else. So that's their cell wall is always cellulose. Fungi will always deposit chitin. Bacteria will always deposit peptidoglycan, which is peptide, a protein, and glycan. So it's glycan. L comes over here. Glycan, which is a carbohydrate combined together on the outside, and then there's a virus which does not have a cell wall. It can have a cell wall as well, which is made up of proteins. It does actually have a cell wall. It doesn't have a virus structure is very different. It doesn't have an exact membrane. Its wall is a cell wall which is made up of proteins, and then it can be further covered by a slime capsule. So just to give you guys an example, there's gram-positive and gram-negative bacteria, in which, look at this, deposition of that peptidoglycan cell wall. So it can be either a complete deposition, or it can be a deposition followed by an outer membrane. So cell wall can be variable, it's not consistent, but one thing to know is that these substances are consistent. And I don't think you need to know the details of this much. And the bacteria chapter has essentially been excluded right now. If in any specific MDCAT it is included, then we will do these in detail at that point.